The second type of horizontal gene transfer is transduction. And transduction occurs naturally in a broad spectrum of bacteria, and we'll actually see why. But they must be of the same species for the most part, okay? And that's because bacteriophages, the viruses that affect or infect bacteria, usually only infect the same species. There's a little bit of question about that, but for the most part, if a virus can infect E. coli, then it will only infect E. coli. But then there's the transfer of DNA from one bacterium to another by that bacteriophage. And the bacteriophage are more numerous than all of the bacteria in archaea. So we never really think about viruses infecting other living things besides, for example, coronavirus infecting us now. But viruses infect every living thing, whether they be plants or protozoan, or in this case, bacteria and archaea. So there's two types of transduction. And the first one is generalized transduction. And that involves a virulent phage. And a virulent phage means a phage that is going to break apart the bacterial cell when it's done, and it will never incorporate itself into the bacterial genome. That'll be a little confusing right now until we really get to talk about viruses. So the phage, and it's phage or phage, bacteriophage, doesn't matter to me how you say it, but remember that it is a virus, and it attaches to the bacterial cell and injects viral DNA. The host cell, and I've got a slide of this after, and the host cell DNA is going to be broken up by phage enzymes. The virus needs a bacteria in order to use its cellular processes. But what it does not want is it doesn't want the competition for making proteins for the bacteria to interfere with making new viruses. So one of the first things that the virus is going to do when it infects the bacterial cell is it's going to chop up the host DNA. So the phage DNA is now in control, and it's going to direct the cell to synthesize viral components. So the parts of the virus are going to be made by the bacterial cell. So the ribosomes are going to be important. Also the DNA polymerase, maybe the RNA polymerase, to make more of the viral genome. So that's going to be important. But then we're going to get to a point where we've got all the pieces assembled, and we need to, or we've got the pieces made, now we need to assemble them into new viruses. So during that process, the new genome is going to be put into the virus. And so when that virus is packed with the genome, sometimes that fragmented bacterial genome is going to be inserted either with or instead of that phage DNA or phage genetic material. And when that bacterial cell lyses, it's going to release all these new infective phages. If at that point, one of those infected phages that has now the bacterial DNA goes to a new bacteria and inserts that new genetic material into another bacteria, if that bacteria is not destroyed, then there is new material, new genetic material that has been inserted in that bacteria. So and I just explained number 6. So this defective phage, which we also call a transducing, because transducing means carrying. So this is the carrying virus. is going to inject that bacterial DNA. So here's our virus. This kind of looks like a spaceship or something. These are unusual looking viruses compared to our human viruses. But the head here is where the genome is being carried. And these are tail fibers. And this is a shaft, and this is hollow, and it will inject this DNA down into our bacterial cell, which we recognize here. And one of the first things, there's also, along with this genetic material, frequently there's at least 1 or 2 proteins that go with the genome that are in the head. And they'll go with the genome. So one of those is going to start by breaking up the host DNA. All of these pieces now are going to be made and reassembled. Remember, the virus is going to co-opt all the machinery. Basically, the ribosomes are going to be our biggie, right? And they're going to co-opt those ribosomes to make and control making new parts, new proteins. And then they're going to be put together, and this is a transducing phage. This has a different genome now. This is a DNA genome. And when this comes in and it brings in a different DNA, right? And remember, that new DNA doesn't have the same goal in here. It doesn't code for the same thing. It doesn't code for new virus. So now this is going to be bacterial DNA, and that bacterial DNA will become incorporated into the bacterial genome. And that's called transduction. And that leads us to talking a little bit about plasmids, because plasmids are one of the ways that we actually send DNA to other places. Okay, plasmids are a ring-shaped, double-stranded piece of DNA that is self-replicating. It is not incorporated into the chromosome of the bacteria. So when I say it's self-replicating, the bacterial chromosome doesn't always replicate. It will replicate when it wants to undergo binary fission. But the plasmid can replicate when it wants to. Okay, it does not, it's not under control of the chromosomal DNA replication cycle. And it carries genes that are not essential for the cell's survival. That is, in most cases. Every once in a while, you'll have a plasmid that may be essential for the cell's survival, but usually the plasmids are not. So there are different types of plasmids, and the ones that we most commonly will see are called an F plasmid. And an F plasmid we cover here specifically because it directs the synthesis of a pilus. And that pilus is required for transfer of chromosomal genes. And our next method of horizontal gene transfer, called conjugation, is going to require an organism to have that F plasmid in order to make the pilus. An R plasmid codes for antibiotic resistance. We have virulence plasmids, and those can code for toxins or certain enzymes that give the organism a selective advantage. And then we have bacteriocin plasmids, which are kind of little buggers because they kill other bacteria. And you kind of think, well, why would you want a plasmid to kill other bacteria? But remember that bacteria compete for resources as well. So if in certain circumstances it may be advantageous to have your own area and not, for example, have to compete with other bacteria for limited resources. Now, plasmids can qualify as naked DNA. Okay, so they can be transferred by transformation, they can also end up in the DNA of a virus, right? Or as the DNA that a virus carries. And they can also be passed through conjugation. And so let's see how.